Aloha, this is Roger Brewer with the Hawaii Department of Health. This is a recording of a presentation entitled Improving Data Reliability and Minimizing Delays with Incremental Soil Sampling. I originally gave this presentation at an Associated General Contractors Conference in Arlington, Virginia in September 2015. The target audience were construction workers or large redevelopment projects that often deal with contaminated soil. But really the problems we talk about are common to any environmental investigation. So what I'll talk about today has to do with sampling theory. And one thing to point out is sampling theory is independent of the media that you're testing. Whether it's people, food, pharmaceutical, surface water, it all comes down to the same basic problems. So how do you collect a representative sample of something you're trying to test? We just happen to be talking about dirt. Quick outline, we're going to discuss what we've been doing for the past 30 plus years with discrete soil sampling, why it's unreliable and inefficient, and why we should care, and why a switch to incremental sampling is necessary and how it's done. Just to quote our friend Albert Einstein, if I had 1 hour to solve the problems of the world, I would spend 59 minutes on explaining the problem and 1 minute on explaining the answer. That's more or less what we're going to do. We're going to focus on initially problems with discrete soil sampling and why these problems occur, and then we'll talk a little bit about a solution in terms of decision unit and incremental sampling methods. So what's wrong with my discrete soil sample data? This is something that often comes up at redevelopment, large construction projects, or any environmental investigation. There's very often a need for multiple remobilizations and step-out investigations with no really clear endpoint. It's very common to have failed confirmation samples after you thought you identified the extent of contamination in an area, but find out otherwise, and you collect confirmation samples and your excavations seem to grow larger and larger over time, again without a clear endpoint. A big issue that we've been dealing with here in Hawaii, I'm sure it's common in other places, the accidental import of export of contaminated soil to other properties or to your property that's being redeveloped. Here's an example of discrete sample investigation. In this case, I've got 25 discrete sample points scattered across an area we suspect might be contaminated. And it's a very common question, how many discrete samples do we need to characterize an area? But there's really no science behind use of discrete samples to begin with. And the basic answer to that question is you take the budget that's allocated to laboratory analysis divided by the cost per analysis, and that's how many samples you can afford to collect at a site. That's really what it comes down to in the field more often than not. But in this case, we've laid out our 25 grid points to collect discrete samples, and you can see here the X's that are red identify areas that are above our hypothetical screening level of 1 ppm. Yellow means the contaminant was detected, but below that concentration, green, it wasn't detected. You see a very common pattern of parent contaminant distribution when using discrete samples. You have a core area of contamination and these isolated apparent hot spots around it, where maybe you think contaminant was spilled outside of the main area where it was being used. And you see these odd apparent cold spots within contaminated areas. And it's understandable to think maybe nothing was going on here, it just happened not to be contaminated during the operations. But the first flag that pops up here is we're comparing these individual discrete soil samples to published screening levels. One thing to keep in mind is the screening levels published both by Department of Health in Hawaii and also by US EPA, their regional screening levels, were never intended to be applied to individual discrete sample points. If you look at how those screening levels are developed, they're intended to be applied to the average concentration over some targeted area, like a targeted exposure area or even a targeted spill area, not the individual points. So that's the first flag that something may be wrong here with the approach we're using. And nonetheless, let's continue on here. So in this example, we've excavated out the contaminated soil that was identified during the initial investigation, and we collected confirmation samples around the perimeter of the excavations. Not too surprisingly, some of the confirmation samples that we thought should be in clean areas come up hot or above our screening level. So the next step, we excavate these areas, so additional over excavation, collect additional confirmation samples, and then see what you get. And as many of you I'm sure know, this can go on and on, again with no clear endpoint in sight. if you're dealing with an active construction project, this can be a complete disaster because they've got their budgets already in line, their construction contracts in place and such, and any types of time delays in particular can really mess up an entire project. So it adds significant time and cost to the project. And really time is the bigger factor than cost in a lot of cases. So what's going on here? Maybe it has something to do with the way we're collecting the samples. And anyone who's ever done any field work probably wonders the same thing. What if I move my sample point over a few feet? Would I get a different answer, and how different would it be? Or what if the lab tested a different aliquot of soil from the discrete soil sample I sent, or a different spoonful or pinch of soil from that jar? One thing to keep in mind when the lab tests soil for metals, they're only testing about 1 gram of soil. That's a pinch. VOCs are only testing about 5 grams. PCBs, pesticides, dioxins, TPH, heavier contaminants, they're testing 10 to 30 grams, so maybe a spoonful of soil. And you might think the labs homogenize the sample. You really can't homogenize the sample. If anything, you can make it worse as fine grain material settles down to the bottom that could be more heavily contaminated. And if they only test the top part of the sample, then it's not going to be representative of the sample at all. Well, we wondered the same thing. So 2 years ago we went out to 3 different sites in Hawaii and collected hundreds of discrete samples at each one of these sites. The first site was an area known to be contaminated with arsenic from wastewater from a former industry. The second site was an area known to be contaminated with lead from incinerator ash that was mixed with fill material. The third site was an area known to be contaminated with PCBs from past releases of waste transformer oil. Each one of these sites we set up a 24-point grid. In each one of the grids we collected multiple discrete samples and tested each of them. This study is published in a document called Small Scale Variability of Discrete Soil Sample Data, and you can see the web page where it's posted at the bottom of the slide. Now at each one of the grid sites, the grid points, we did 2 things. First, we tested for inter-sample variability. This is the variability between co-located samples. In this case, around each one of the grid points, we collected 5 discrete samples, 1 in the middle and 4 at each corners, within a 3-foot grid, 3-foot square around the grid point. The discrete samples were 200 to 300 grams. Key point here is for each one of the samples, we had them processed at the laboratory using incremental sampling methods, where they were dried, sieved, and subsampled for testing, so we can be highly confident that we have good data for each one of these samples. Second thing we did is we looked at the variability within a single sample, or intra-sample variability. In this case, for metals, we took the center sample collected from each grid point, and we tested it 10 times with a portable XRF without disturbing the sample when it was collected. For PCBs, we collected a 6th sample at each grid point. In this case, we took the sample and put it into 10 separate jars and had the laboratory test each one of these subsamples. Well, here's what we found. This is the PCB site where we saw the most variability. In this case, we're looking at the inter-sample variability of co-located samples around 1 point. You can see the lowest concentration of PCBs identified in the grid point is 4.9 ppm, but just a foot away we had a concentration of PCBs in one sample of 91 ppm. Also, you see this 2400 ppm. Keep an eye on that. That was the center sample that we had tested 10 times. Let's look at it. So if on average for the inter-sample variability at the PCB site, we saw 10-fold variability around each of the grid points just between the co-located samples. Maximum variability around any single grid point, almost 500-fold. Let's look at that center sample again. In this case, we tested one sample 10 times. You see that the range in PCB concentrations within the sample, with the low of 810 ppm, high was 5700 ppm. So this is within a single sample, almost an order of magnitude. We saw an average variability between samples that we tested here of 17-fold. It didn't really matter what the concentration was. We saw the same variability within samples, whether it was high concentration of PCBs or low concentration of PCBs. Again, the maximum variability that we saw was 1600-fold, or over 2 orders of magnitude within a single sample. In this particular sample, at this grid point, the average concentration was 2400 ppm. So what's going on here is we looked at the soil sample under more detail in a microscope. One thing we noticed was these small dark colored nuggets, and they're very different from the rock fragments in the soil. These nuggets were easily broken. If you zoomed in on them even closer in the upper left hand corner photograph, you could see what looked like eggs almost, and there was a very distinct rim around the outer edge of them. But if you look closer, the inside of these nuggets are filled with a dense fine-grained material and very dark colored. So we suspected what these were PCB infused tar balls within the soil. If you happen to get one of these in one of your discrete samples, and your reported PCB concentration would be very high. If you didn't, then it wouldn't be very high. So it's completely random in nature. Well, a chemist friend who likes to cook showed me how to make these nuggets myself. We took some dry flour and dropped olive oil on it. The olive oil very quickly will beat up on top of the dry flour, and over time the olive oil would slowly sink into the flour. And one thing you see in the lower right hand corner, very distinct rim around each one of these nuggets as they sink into the flour. Then we took a sieve and ran the flour through it, and you can see there's lots of very tiny olive oil infused nuggets in the flour. Again, if you were testing this flour for olive oil, if you happen to get some of these nuggets in your sample that was actually being tested, you'd see very high concentrations of olive oil. Otherwise, not much. This is how PCB nuggets probably form in soil. It's from releases of PCB oil onto dry, fine-grained soil. They sink into the soil and form small nuggets. If you get a nugget again in the aliquot tested by laboratory, you get a high concentration of PCBs. Otherwise, you may not. And again, it's totally random. So no wonder discrete samples don't work. And the bottom line is they're just too small. They're too small to capture small-scale random variability in the area where you collect the sample. And again, keep in mind the mass tested by laboratory. It's very small. These masses aren't based on sampling theory or anything really have to do with sampling. They're just the mass that the laboratory needs to test it with their analytical equipment. Now, if you could see contamination in soil, it'd probably look like something like a Jackson Pollock splatter painting. Here on the left, the blow-up of his painting there, that red dot is actually the size roughly of a discrete soil sample that might be collected there, about the size of Pollock's foot. Now imagine moving that red dot around his painting, and you get highly variable and very random concentrations of dark versus light, just depending on where you happen to place that discrete soil sample. On the right hand photograph is a picture of a milk truck that was flipped over. You see how heterogeneous the milk spread through the soil. The red dot again represents a single discrete sample, and imagine trying to map out the contamination with discrete samples here. It'd be completely random. You'd find some spots and you'd miss others, but if this was invisible, you'd really have no clear idea of what the actual contaminant distribution looked like. So that's the bottom line. You can't really rely on discrete soil samples to map out areas of contamination, or even to estimate concentrations of a contaminant around any single point or for a given area. So this explains why confirmation samples fail or even pass in the field after you've excavated an area of contamination. And in the end, it's really random. It's due to small-scale random variability of contaminant concentrations, just over a few inches or a few millimeters or feet. The concentration reported for any given discrete sample is largely random. It's highly unlikely that concentration represents the average concentration of the contaminant in the sample. It's highly unlikely that that sample actually represents the typical or average concentration of the contaminant from where that sample was collected. So if you look in the right hand corner of the figure here, in that one corner where a confirmation sample was collected, if you move the sample point over just a few inches or something, you could get a completely different answer, and you could get multiple answers for any given area if you collected lots of confirmation samples or discrete samples and tested them in a highly random area. So how this happened? Here's two quotes from some guidance published in the mid-1980s by US EPA. First, for the guidance verification of PCB spill cleanup. This was published at the same time TSCA regulations were being developed. So the implicit assumption that contamination is likely to be uniformly present anywhere within the sampling area is reasonable. So in other words, you get the same concentration within an area, a spill area, regardless of where you collected the sample. The second is when there's little distance between points, it is expected that there will be little variability between points. It's from EPA 1989 guidance methods for evaluating the attainment of cleaned up standards. This is a highly referenced document. The problem here, if you look back at the origins of these guidance documents, is they were developed really prior to a time that we had a lot of experience testing soil and understood the nature of small-scale variability. The guidance really reflects experience with industrial waste streams. So picture a pipe coming out of a factory, and here the concentration of contaminant in the waste stream is going to be very consistent as long as the process inside the factory remains consistent. So you can collect a sample at any time and any mass of sample, you'll get roughly the same answer. And the thought was at the time that should be the same case for soil. A big issue was the use of discrete soil sampling methods was never tested in the field while the guidance is being prepared. Also, there's a lack of training in sampling theory at that time. It's unlike the mineral exploration business and agricultural businesses where sampling theory was becoming very popular. Also, another big issue is that field quality of environmental data, unlike, say, the mining business or agricultural business, isn't market driven. So there's not a lot of impetus to go back and test your data or test your results compared to these other industries. Well, the solution, in the end, you go back to sampling theory. And there's two common themes we're going to talk about: decision units and incremental sampling. Someone sent this quote from Dr. Zeus: "Sometimes the questions are complicated and the answers are easy." Really, that's the case. You may have joined in some workshops on sampling theory and it can seem really complicated, but in the end it's actually very simple. Understanding the nature of heterogeneity in soil can be complicated. It takes a while to think about, but addressing it as part of field investigation is simple. So there are two key parts in sampling theory, two key terms we use: decision units and incremental sampling. First, decision units. The strict definition of decision units: an area or volume of soil about which a decision will be made. In the end, we've always been doing this. Decision units are areas in the field that are designated based on suspect spill areas, or areas you suspect to be more heavily contaminated than others, or they could be designated based on an exposure area, say someone's backyard or a large commercial property with a lot of exposed soil. So the objective is to determine the mean concentration of the contaminant for this decision unit volume and area of soil. It's the basis of risk assessment and risk-based screening levels that we talked about before. A key point, though, is we know now that you can't just collect a single sample within one of these areas. It's not going to be representative. The key point is that the sample that you collect has to capture and represent small-scale variability within the decision unit. Now, easy definition of a decision unit, again, to think about is it's the volume of soil that you would send to the laboratory as a single sample if possible. So let's say we could dig up our entire backyard, send it to the laboratory for testing as a single sample, then we get a single number. We can't do that, so we have to collect a sample from that targeted area. Again, this approach has been used in mining, agriculture industries for decades. Now, within each one of these decision units, we're going to collect a single incremental soil sample, or a multi-increment sample as it's referred to in Hawaii. So in this case, we're going to collect a sample by combining soil from 30 to 75 points within the decision units. These are small increments of soil, 25 to 50 grams each. We'll combine all these together to prepare a 1 to 2 kilogram bulk sample for that decision unit. This is kind of similar in concept to composite samples, but there's some very important differences we're going to talk about later on when we discuss issues with TSCA. A big issue here is the samples are carefully processed and subsampled at the laboratory after they're collected to retain the field representatives. You're not just going to open up the bag or the jar and test a random spoonful of soil from the sample. Also, replicate samples are collected from different areas within the decision unit. and then each individual sample is tested, and then the results are compared to test the precision of the data. Again, the objective is in these investigations to identify hot areas in the terms of a risk sense and not hot spots within the DU. You will make sure that your sample you collect adequately captures representative number of hot spots and cold spots within the DU, but ultimately you're after the mean concentration. As always, the issue that comes up is, well, what about acute toxicity from one spot, one of those little X's in the figure? And in acute toxicity, you might think of 10 grams of soil. That's the mass of soil assumed that a pica child or a kid that eats dirt might be assumed to ingest at one time. Well, first point is there's no acute toxicity has never been documented for exposure to soil. Usually we're talking about contaminant levels in the parts per million or very, very low concentrations. Also, there's no acute toxicity factors for the large majority of contaminants we deal with, so there's no acute toxicity at soil screening levels. They're all based on chronic toxicity or long-term exposure after many, many years. Also, if you think about it, it would be impossible to verify that no single spoonful of soil within some targeted area exceeded some hypothetical acute screening level, even if you could develop one. Another issue that comes up is what's the maximum concentration within the decision, and really this is a leftover from the idea of discrete samples. Again, if you think about the maximum concentration of a contaminant within any area, if it's there, the maximum concentration is always going to be 1 million parts per million at some scale. If you could go in and test that area with a pair of tweezers and pluck out a single particle and test it, eventually you would find a concentration of 1 million parts per million. So the whole idea of maximum concentration was really a fallacy. It's completely arbitrary based on the mass that happened to be tested by a laboratory. It never really had anything to do with risk. So the basis of incremental sampling is really based on a sampling theory, and there are lots of causes of erroneous data or ways you can mess up your sample data: fundamental error or the mass of contaminant of soil collected. You see lots of different errors here. We're not going to go into each one of these, but I want to explain sampling theory in another way here in a minute. If you want to get the details of sampling theory, the first thing to do would be to take Chuck Ramsey's 4-day class, where he goes into a lot of detail about sampling theory, all the different types of error we looked at a few seconds ago, and he goes over the collection of incremental samples, or multi-increment samples as he calls them, as part of site investigations. If you want a quick introduction to incremental sampling and sampling theory, then check out ITRC's incremental sampling methodology webinars. They're taught several times a year. You see the web page there below. They're in 2 parts, 2 hours per part. To date, over 3,000 people have joined in these webinars. Now, for carrying out incremental sampling in the field and designating decision units, right now Hawaii's guidance is the best out there, our technical guidance manual. There's 3 sections in there that discuss implementation of DU MIS investigations in the field. You see the web page below, and that guidance is currently being updated. We hope to have it posted by the end of year 2015. So rather than get into details in sampling theory, the theoretical part, I want to explain it using a salad. In the end, it is really simple. Let's say you had a large salad here. Your primary concern was exposure to toxic tomatoes. Maybe you're allergic to tomatoes or something. But here's the wrong questions to ask. What's the maximum concentration of tomato in the salad? Well, that's an easy question. It's 1 million parts per million if you happen to pluck out a single piece of tomato and test it. Or what's the concentration of tomato at point X? That's not really the point either. The primary concern here is long-term exposure to tomatoes in the salad over many years. So really the first step to address this problem in terms of sampling theory is to define your investigation objectives and designate decision units. So the right question here is, what's the mean concentration of tomato in this salad? Again, the primary concern is long-term repeated exposure to tomatoes or contaminants in soil. In this case, the salad we're going to assume would be eaten over a lifetime. The entire salad represents a decision unit or an exposure area decision unit in terms of sampling theory and risk. So the next step is to collect a representative sample. And again, in this case, we're going to collect a really big sample in order to capture and represent small-scale random variability within the salad. Now for soil, like I mentioned before, Typical sample size is 1 to 2 kilograms, roughly 1 to 2 gallons of soil. Here's how we're going to collect a representative sample. We're not just going to go to a single point and grab a handful of sample. The sample is collected through it by grabbing small increments of salad over multiple points across our targeted decision unit. So again, the sample, you have to collect enough points to capture a representative number of hot spots and cold spots within the salad. If this was soil, we'd collect increments of salad from at least 30 points if we assume it's low heterogeneity, or up to 75 or more points if we assume there's high heterogeneity, say tomato distribution within the salad. Now when the laboratory gets our really large sample, then again they're not just going to grab a handful and test it. If this was soil, the bulk soil sample will be dried, sieved, and subsampled. In the case of salad, this example here, they're going to collect small amounts of salad from, again, 30 or more points within the salad, repeating the same process in the field. And then the subsample that you collect is going to be representative of the sample that was sent in, and the sample that you sent in was representative of the area in the field. And that subsample is the aliquot tested by the laboratory. Again, if this was soil, then we'd be testing a minimum of 10 grams. Another thing we're going to do is we're going to collect 3 different samples. We're going to go back and completely redo our sampling 3 times from independent areas, so we come up with 3 different samples, 3 different analyses. And if we did this right, then our analysis should be roughly similar. Now if needed, then you can increase the resolution of data. If, let's say, assume you're pretty confident that if you tested this all as a single decision, it's probably going to fail because there are a lot of tomatoes in there, but you suspect that most of the tomatoes in one area, when in the field, in case of soil, this would be a suspect spill area. You cordon off this area as a separate decision unit for characterization, and then you would add decision units on either side of this area. We refer to these as perimeter decision units. We anticipate these being clean. This is a useful approach to take at a site, is focus on what you assume to be spill areas and try to clear other areas around it. If a site's under construction, this would allow large parts of a site often to be quickly cleared so work can continue, and then work would focus on small areas of the site where you did identify heavily contaminated soil, or in this case salad. Now compare this to discrete samples. Let's say we collected 6 discrete samples here, random, within the salad, just small handfuls. Really, with a discrete sample, it's like testing a salad with a pair of chopsticks. It's highly random, the concentration of anything you get within a single spot. So this discrete data really wouldn't be that useful for testing a salad. They're really not that reliable either for testing soil. You may find something and think you've landed on isolated contaminated areas, where it is in reality it's just confused with small scale variability within the salad. You really don't have a clear idea of what's going on. More food for thought. This is another example we gave out, used in other workshops. Here's what we thought in the 1980s, that contaminated soil is like a bowl of Cheerios. So the same thing, any small sample you collect from that bowl, you're going to get the same answer, so it doesn't really matter where you collect the sample or even the size of the sample you collect. So what we now know in the 2010s is that contaminated soil is more like a bowl of Fruit Loops. So the contaminant concentration can vary very dramatically just between adjacent points. And the only way you can get a representative sample from a bowl of Fruit Loops or something like this is to collect a large sample from a lot of different points within your targeted medium. So let's go back to the example we started with here, and instead of discrete samples, now we're going to use a decision unit incremental sampling approach to investigate this area. Let's assume that the center part of this area we have good reason to suspect is heavily contaminated with arsenic. We're fairly confident we can draw a clean line around it. So within the heavily contaminated area, we designate decision units for spill area decision units. And typically in the field, spill area decision units range in area from a few hundred square feet to a few thousand square feet. And around these spill area decision units, then we designate perimeter decision units in areas that we anticipate are going to be clean. So we have 4 spill area decision units and 6 perimeter DUs. Again, the objective is to determine the mean concentration, in this case an example arsenic, within each one of these decision units. Now instead of collecting a single discrete sample within these decision units, we're going to collect a single multi-increment sample, again hitting 30 to 50 points. And we're combining 10 to 30 or 50 grams of soil from each point into a bulk sample for that decision unit. The bulk sample is going to weigh 1 to 2 kilograms. Within 10% of the decision units, or in this case 2 of the DUs, we're going to collect triplicate samples. So we're going to go back and collect 2 completely independent multi-increment samples within 2 of the DUs. We have 3 samples for each of 2 DUs. Then we can compare the results and get an idea of how representative our samples are. This example, in the replicate data, samples A, B, and C, 149 ppm, 179 ppm, and 135 ppm arsenic. So the relative standard deviation 16%. If it's under 35%, that's pretty good. If it's between 35 and say 100%, it's maybe usable, maybe not. If once you get a relative standard deviation over 100%, then it gets to be really questionable if your data are usable at all. In this case, we collect a multi-increment sample from each one of the decision units. And on average, once you get these set up, it takes maybe 10 to 20 minutes per sample to collect. And we're going to send 16 samples to the lab now instead of 25 like we did in the discrete sampling approach. So less samples, maybe a little more time in the field to set it up. So here's the example results. In this example, the middle part was heavily contaminated as we anticipated. One area was under the screening level, but there's still some contamination there. Did a good job designating clean perimeter decision units around 3 of the edges of the site, but on the edge of the site to the right hand corner, 2 of the decision units in this example turned out to be contaminated. We anticipated they were going to be clean. This is going to require another round of step out sampling, but in this case we'd probably designate 2 rows of perimeter decision units along this area where we're highly confident that the outer row is going to pass, if not the inner row. Also in this area, once we define the lateral extent of contamination, we go back into this area, and each of the DUs, we may combine them in larger DUs, but we would drill through it and collect multi-increment samples at depth to determine the base of the contamination. This is done before you even excavate the site. So when you do go back and excavate it, you already have your confirmation samples. Well, there are a lot of details to doing this, obviously. In our guidance, we go over different types of tools to deal with different types of soil in different depths, and how the shape of increments should be collected cylindrical, not wedge shaped and such. Also, we talk about designation of decision units for ecological risk. In this example is a skeet range. You may have spill area decision units or source areas. It's called in ITRC's guidance in areas where you think the skeet may have fallen, and that's surrounded by ecological based decision units based on the ranges of birds or other animals that you may be using in your ecological risk assessment. You can do this also for sediment. You use the same approach we discussed a minute ago for excavating sites. If you already excavated an area, then the bottom and each side of excavation could be considered individual decision units. Or again, you could drill this ahead of time, determine the base of contamination and the lateral extent of contamination before you excavate, and that negates the need to collect confirmation samples after you've done your excavation, so you get in and you get out. Same thing with stockpiles. In this example, you can assume a certain volume of soil for, say, a residential site, 100 to 400 cubic yards. That's enough soil to cover 5,000 square foot, say, exposure area to a depth of 6 inches. So in this case, you would spread the stockpiled soil out into a flatter layer, no more than a few feet thick. Break it up into your target volumes, your decision of volumes, and collect a multi-increment sample within each one of these targeted DU volumes of soil in your stockpile, and make a decision DU by DU as to where the soil can be reused. Now, decision of volumes for a commercial site is probably going to be a little bit larger, 400 to 800 cubic yards based on half-acre exposure areas, and they can be larger than that depending on what your reuse is of the soil. You can do incremental sampling for subsurface also soils. In this example, they dug a ditch or a trench through a site. They did standard surface incremental samples for the surface soils. Along the edge of the trench, it designated specific layers of soil for testing, and it collected an incremental sample along the exposed layer of the soil within the trench. Again, do this, you collect incremental samples for subsurface soils. Just because there's soil on top of the targeted layer that you want to characterize doesn't mean you can revert to discrete sampling. In this case, you may put in a few test borings at a site, determine how to break it up into targeted decision unit layers. And then each core through that layer represents an increment. Ideally, you'd install at least 30 cores through each one of your targeted DU layers. Now, the cores are going to be too large to combine into one large sample, so you normally have to subsample the core increments for each decision unit layer, and then combine those into a sample to be tested in laboratory. Again, that's discussed in our guide. In some cases, you're just not going to put in 30 borings for various reasons at a site, either access or the cost or whatever. But if you're going to rely on single borings to make decisions, you still wouldn't collect discrete samples from the borings at targeted depths. What you would do is you take the core and break it up into targeted depth intervals, say 0 to 1 foot, 2 foot to 5 foot, 5 foot to 10 foot, and such. And then you'd send that entire length of the core to the laboratory for processing. So they would test the entire decision unit layer instead of individual discrete samples scattered through that. That avoids the need to extrapolate between data gaps. Same thing if you were trying to test the depth of contaminated fuel at a specific spot at a site. You put a boring in there and divide up the core into targeted decision unit layers rather than look at individual depths for discrete samples. This type of investigation, you're only relying on a single boring, a small number of borings. You can't really trust the concentration you get, but it'll give you a good idea whether contamination is present or absent. Now you can do incremental sampling for VOCs. You just take a jar of methanol in the field with you and put subsamples from your increments into the methanol, or from exposed sidewalls or trenches or such, and directly into methanol. That's discussed in our guidance. Now, one question that came up during the conference in Arlington was the use of incremental sampling for PCBs under TSCA. That's something we've been dealing with for quite a long time. Now, thinking back, we already discussed that, but in the 1980s guidance, the assumption was that a single discrete sample was adequate to represent what they called a sampling area. Again, quoting their guidance, the implicit assumption that contamination is likely to be uniformly present within this sampling area is reasonable. But we now know that's just not the case. And the diagram in the upper right hand corner is an actual figure from that TSCA guidance. So we can't really rely on single samples to characterize a sampling area. Now, what they refer to as sampling area, or essentially match the intent of what we now refer to as decision unit. We have a much better idea of how to designate decision units in terms of suspect spill areas or health risk in terms of exposure areas. We know the incremental samples are required to get a representative concentration of a contaminant within a targeted area. One key point under TSCA, they have a lot of discussion against dilution of contamination. You're testing for PCB contamination in soil. In an incremental sampling approach, a decision approach, the intentional combination of anticipated PCB spill areas with anticipated clean areas as a single DU for testing could violate this anti-dilution clause in TSCA. And here you can see it states no person may avoid any provision specifying a PCB concentration by diluting the PCBs unless otherwise provided. So that's a key point when you're investigating PCB releases, is you need to coordinate with the overseeing regulatory agency, in this case our office, Department of Health, or in some cases EPA Region 9 may be involved, to very carefully designate spill area decision units across that property or that site, just to ensure that you aren't intentionally mixing spill areas with anticipated clean areas. Another thing that's allowed under TSCA is limited compositing of discrete samples. In this case, their guidance allows compositing up to 10 different discrete samples from, say, 10 different discrete sampling areas as a single sample. They did this just to help reduce the costs of laboratory analysis. In this case, though, your laboratory data had to be divided by the number of discrete samples, or more specifically the number of sampling areas that were included in that composite for comparison to a cleanup level. This was to ensure that no single sampling area exceeds a target cleanup level. Now, rethinking about this in terms of decision units and incremental sampling, this would be the same as collecting a multi-increment sample from each of several different decision units and then combining all these samples into a single sample for testing, and then dividing the resulting laboratory concentration by the number of decision units that were included in that composite incremental sample. Well, compositing of incremental samples or compositing of samples across DUs isn't allowed under Hawaii guidance or even under ITRC guidance. So that's a quick answer to the idea of compositing for incremental samples. It's just simply not allowed. Instead, you have to designate decision unit sizes up front that are adequate for decision making. You're required to collect a separate incremental sample from each one of the decision units and independently test that sample and get a single concentration for each decision unit. You also want to make sure you include an adequate number of increments for each one of these decision units, and to prepare a bulk sample that's at least 1 to 2 kilograms in size. And then again, each sample is independently processed, subsampled, and tested at lab, and an independent decision is made for each one of the decision units. So we're still working with the US EPA on a pathway forward, a regulatory pathway forward, using the risk-based option under TSCA to incorporate incremental sampling and decision unit methodologies. The technical justification need to switch from discrete to incremental samples under TSCA is largely agreed on. We just need to work out the regulatory pathway. At some point, though, we all have to just admit that discrete sampling just doesn't work. It's not reliable or efficient. And we now know the world isn't flat. And incremental sampling is the only way you can get representative data at PCB contaminated sites as well as at other sites. So in summary, discrete soil sample data unreliable, inefficient for use, environmental investigations, something we learned over the last 30 years. Even so, you can use grids of discrete samples that might be useful for identifying large-scale areas of contamination and designating decision units. You just have to be very careful, though. You can be easily fooled by heterogeneity with sort of artificial patterns of contaminant distribution within an area. And again, decision units are based on the investigation objectives, either identifying spill areas or for risk or for optimizing remedial actions. Incremental samples are collected to ensure that you get reproducible, technically defensible data. A question always comes up about cost. In general, the costs work out roughly the same. You're going to spend more time up front designating DUs and understanding the history of the site under a DU incremental sampling approach, but your lab costs are going to be much lower than they would be for a discrete sampling approach. And more important, it's going to be much more cost effective by the project end because you can much more easily designate an endpoint to a project. Also reduces uncertainty in your investigation and potential for unanticipated delays and future environmental liability if you have to miss contamination. But we still need a lot of training and discussions to overcome ingrained regulatory hurdles, especially under programs like TSCA. And in the end, experience in the field is the best teacher. So with that, we ended the presentation at the Associated General Contractors Conference in Arlington, Virginia. And we had some of the questions we went over. I've already covered them in this recording. And we talked about costs. We talked about regulatory acceptance. One thing that we pointed out was that ITRC's ISM webinars now have had over 3,000 attendees. 1,500 of those attendees were regulators, including several hundred from US EPA. So a lot of people are getting more of a basic understanding of incremental sampling and still getting a better understanding of limitations of discrete sampling. As far as how to get this started where you happen to be in your state, if you're not already using it, what we did is we initially started with some pilot studies, and also we spent probably 2 or 3 years just in the transition of moving from our past discrete sampling methods to incremental sampling methods. Projects are already underway based on discrete sample data. We didn't go back and require that new data be collected. But we did encourage that any confirmation samples collected, that incremental sampling methods would be used. A key point here is, from our experience too, is you really need to get the regulators out in the field. We're lucky in Hawaii, we have our own sampling team within the Hawaii Department of Health. I think that's one reason why it's taken off here. So if you're a consultant or you're wondering how to get the regulators more involved, you're a regulator trying to get an idea of how to better understand this, then really the next step is to get out in the field as much as possible. So thanks for joining in, and you have my email address on the first slide. I'd be happy to talk about any details. Thanks.